Listen-only mode.
Ladies and gentlemen, thank you for joining us for today's webinar, HLA-Profiled Cells and Tissue in Preclinical Research presented by AllCells. My name is Afshin Shirazi. I'm Marketing Manager at AllCells, and I'll be moderating today's presentation. Before we get started, please be aware that everyone has joined today's program automatically muted, and in order to participate in the question and answer session immediately following the presentation, please enter your questions at any time using the GoToWebinar prompt at the right of your screen, and we will address them at the conclusion of today's webinar. Today's topic has been selected because we have seen increased interest from researchers on HLA typing and how this can be a critical factor in the assay development, screening, and investigating process. We hope that today's presentation can serve as a resource for you and stimulate discussions between your colleagues and ours to enable your research further. I'm very excited to be joined today by my colleague, Dr. Erin Kelly, Bioservices Supervisor at AllCells. Erin has been a scientist here with us at AllCells for over 3 years. She has a PhD in immunology from Weill Cornell Medical School and has experience working with many different primary cells, including monocytes, macrophages, T cells, lipoaspirate, bone marrow, and blood. Please join me in welcoming Erin Kelly.
Thank you, Afshin. It's an exciting time to be a part of the biotech and pharma community and be able to speak with you about HLA data and its applications. As with past AllCells webinars, our objectives are solely to inform you and educate you, our current customers or prospective ones, in concepts in science and technology that are relevant to your clinical and basic research projects. It is also an opportunity to share the experiences we have with our customers every day here with the greater goal of using these communications to advance basic science, translational research, and human health. Today the topic is HLA, which stands for Human Leukocyte Antigen, a gene and protein family that has garnered the attention of many of our customers. Let's start right off the bat with some hard data. Here's a simple chart showing the upward trend of sales of HLA-specific or HLA-typed tissue over the past 2 years at AllCells. The purple line represents the number of HLA-typed lots sold per quarter. This is an interesting trend by itself, but it becomes even more meaningful when overlaid with another piece of data, the number of different customers that are purchasing primary cells with HLA data, which continues to go up at a high rate. Today, over 50 unique customers purchase our HLA-type primary cells and tissues, supporting an increasing relevance of HLA genetic data in pharma and biotechnology applications. Why is this a surprise, you might ask? What makes this observation unique is that all of these customers are using these products for research applications. HLA data has been traditionally used in the clinical setting only in association with organ and stem cell transplantation. So how is this data being used by those doing research at the bench? Hint, it's not a simple answer. HLA is complicated at both the genetic and protein levels. During my training in immunology, I thought I fully understood HLA and its role in the immune system. However, outside the textbook, in the context of clinical and translational research leading to more and more intricate therapies, I am quite certain that my initial impression of HLA simplicity was off the mark. Due to its high diversity and its immunogenicity, HLA-encoded proteins play one of the most complex, important, and often unpredictable roles in immunology, both in the dish and in the patient. In order to explain how HLA data is being used at the bench, I'll first back up a bit to give a short introduction to HLA genetics, as well as review how HLA-encoded proteins function to influence an immune response. Then we will get to the central question of HLA applications in preclinical research. I will introduce examples of how some customers use HLA information, why others require their source material to be of a specific HLA type. As Afshin mentioned, there will be some time left at the end to answer questions that you can write in during the webinar. So let's start at what could be considered the beginning of the story, the chromosome. HLA genes are located within a superlocus on the short arm of human chromosome 6. Genes in this complex are found in 3 discrete clusters and thus categorized into 3 basic groups, class 1, class 2, and class 3. 8 of these genes, or loci, contribute to the majority of HLA diversity and are named HLA-A, B, C, DRB1, DQA1, DQB1, DPA1, and DPB1. Within each of these 8 loci, there's tremendous sequence variation when comparing sequences between individuals within the human population. Each unique sequence variation is represented by a single allele. Thus, each HLA gene of all 8 HLA genes each have many different alleles. What makes the HLA gene family truly unique compared to the rest of the coding genome is the number of identified unique alleles that have been identified for HLA genes, which in the case of HLA-B, has reached over 3,000 possible alleles. HLA-A and HLA-DRB1 also contain a great deal of diversity with over 1,000 possible alleles each. Hundreds of new alleles continue to be discovered every year for these genes. To get a sense of the magnitude of diversity of this gene, compare this to what you may know about other genes, such as the one that determines blood type, which is referred to as the ABO gene. There are only 3 possible sequence variants, or alleles, within the human population for the ABO gene. For CFTR, an autosomal recessive gene implicated in causing cystic fibrosis, there are only 700 known alleles population-wide, several of which in an autosomal form can cause cystic fibrosis. HLA-B is the most diverse of all human genes in the genome. This variation is known to affect individual susceptibility to infectious and autoimmune diseases, and therefore it is likely that this diversity contributes to donor-donor differences in primary cell experiments. Take a look at some of the allele names for the HLA-B locus to develop a basic understanding of how to look at and interpret HLA data. An HLA allele name begins with an HLA prefix followed by a dash and then the specific gene name, for example, HLA-B. Up to 4 groups of digits, each separated by a colon, are addended to the prefix. The first 2 digits indicate the allele group, in this case 08 or 27. The second set of digits designate the subtype, numbers being assigned in the order in which the DNA sequences have been discovered and determined. Allele groups such as B08 and B27 and allele subtypes such as B0801 and B2725 represent 2 parallel systems of nomenclature that are applied to HLA. These 2 systems reflect 2 independent technologies used to detect differences in HLA alleles. The older system, which is still in use today, is a serotyping method and is only able to identify the allele group. This is often classified as low-resolution typing. Newer methods, such as sequencing, are able to discriminate between individual alleles, yielding a 4 or more digit number, and referred to as high-resolution typing. Like other genes, individuals inherit 2 copies of each HLA gene from each parent. Thus, every individual has 2 alleles of each of these 8 major HLA genes. That equates to 16 pieces of data, as indicated on this HLA typing report for a single individual. Allele data at the single gene level is called a genotype. A haplotype is a combination of alleles over multiple genes that have been inherited and expressed together on the same chromosome. Sequencing data typically cannot be used currently to define haplotypes for HLA loci. High-resolution sequencing data is often referred to as a fingerprint. HLA genes, like other encoding genes, are expressed in the cell nucleus as RNA and translated into proteins. HLA-encoded proteins are often called the major histocompatibility complex, or MHC complex, which are represented here by the blue and green clover-shaped icons. HLA and MHC are often used interchangeably, and I will continue to use the HLA term to refer to this gene and its protein products for the rest of the presentation. HLA molecules derived from the class 1 genes, A, B, and C, are heterodimers encoded by a polymorphic alpha chain and a non-polymorphic beta chain. Class 2 HLA, DP and DPQ molecules are also heterodimers, but are encoded by alpha and beta chains, which are both highly polymorphic, generating an even more diverse set of HLA molecules. Moving past HLA genetics, let's briefly review the function that HLA molecules play in cellular immunity. HLA plays a critical role in helping the immune system, specifically T cells, distinguish the body's own proteins from non-self proteins made by foreign invaders, such as virus and bacteria. After a cell ingests a foreign protein, represented here in red, it is digested by the cell into small pieces called peptides. These peptides are transported to the cell's ER, where they bind specifically and non-covalently to host HLA molecules. An HLA peptide complex with a stable binding affinity is transported from the ER to the cell surface, where it can be recognized by a T cell expressing the cognate or matching T cell receptor. Importantly, a cognate T cell receptor is specific not only for the particular peptide, but is specific for the complex of a particular peptide bound to a unique MHC molecule. This basic principle of T cell biology is known as MHC or HLA restriction because the antigen-specific T cell response is restricted by the context of the MHC molecule. Therefore, a T cell that responds to a peptide presented by one HLA variant will not respond to a different peptide bound by the same HLA molecule. Nor will it bind to the same peptide bound to a different HLA variant, as demonstrated by the gray MHC molecule on the left-hand part of the slide. HLA restriction ultimately allows our immune system to demonstrate even greater control over self-non-self recognition. And by expanding the potential TCR repertoire, HLA restriction allows an enormous capacity for the identification of novel and mutated infectious agents and foreign molecules. As mentioned earlier, HLA has traditionally been relegated to the clinic, and data only applied to transplant indications. However, in the past few years, next-generation sequencing approaches have dramatically improved the speed and cost of genomic sequencing, as well as the accuracy of sequence analysis. Since 2008, the price of sequencing an entire genome dropped from $10,000 to under $2,000 today. Occurring within a similar time frame, in the last decade there has concordantly been a surge of applied immunology research, as the immune system continues to be implicated in almost every category of disease. Its role once relegated to protection from exogenous pathogens, defects in the immune system have now been implicated in the causation and pathogenesis of diseases ranging from gastrointestinal health to prostate cancer. Over 50 of AllCells customers purchased cells or tissue with HLA genetic data. When we asked you, our customers, what your applications were for HLA genetic data, there was a wide range of responses, from companies manufacturing research detection agents to basic research and inflammation to development of GMP cellular manufacturing protocols. Although we won't be able to touch on all of these examples in this presentation, we selected 3 types of application to explore in greater depth. The first application is tumor immunology, which broadly represents all stages of research and development in cancer immunotherapies. The second application to be addressed is disease mechanisms, in particular how HLA is implicated in autoimmune disease. The last application for HLA, which I will present on, is in preclinical drug development. Tumor immunology is the study of the interaction of cells of the immune system with tumor cells, a field which has swelled in size with the recent success of novel cancer immunotherapies. Tumor cells function similarly to the infected cell described earlier, breaking down and presenting protein antigens at the cell surface in the form of an HLA peptide complex. Current TCR and CAR T cell therapies are designed to exploit this by creating artificially engineered T cells that can recognize and bind to selective tumor antigens that are highly expressed intracellularly or on the surface of tumor cells. In order to engineer the tumor-targeting T cell, shown in purple on the screen, endogenous T cells are isolated from the patient's blood. DNA encoding a T cell receptor specific for the selected tumor antigen or peptide is transduced into the cells. The engineered T cell are then reinfused to the patient where it should target and kill tumor antigen-expressing tumor cells. Hundreds of TCR and CAR T cell therapies based on a handful of well-characterized tumor antigens are currently being evaluated in clinical trials or in late-stage preclinical trials with varying levels of success. So what does this all have to do with HLA? Simply put, the success of these therapies depend 100% upon the HLA repertoire of the patient. Since TCR recognition is specific to both the cancer antigen and the HLA molecule presenting the peptide antigen, the HLA molecule is critical. Researchers developing TCR therapies must select a tumor antigen that will bind strongly to the T-cell receptor as well as bind to a particular HLA allele. And as we know, these alleles vary immensely in the human population. So how do drug developers select which alleles to incorporate into their design of TCR therapies? The answer is allele prevalence, or frequency, of alleles in a particular population. Here's a chart of the most frequent alleles we have identified in the 350-member subset of our donor pool located in the Bay Area. Percent frequency indicates that the percentage of total donors that express that particular HLA allele. Some of you may not be surprised that A0201 is at the top of the list, expressed in 37% of our donors, a frequency 50% higher than any other HLA variant. A02 and A0201 typed cells and tissues are by far the most requested HLA type by our customers working on TCR-based therapies. The reason for this is obvious. If their therapy is approved, there will be much larger patient base eligible for the therapy than if a lower frequency allele was targeted. Despite the high prevalence of A0201, the pharmaceutical industry is diversifying their TCR therapies under development to target other high-frequency HLA variants. NY-ESO, a well-characterized tumor antigen that has been the target of monoclonal antibody immunotherapies for over 10 years, is now the target of several promising engineered TCR therapies designed to treat melanoma and synovial sarcoma. Hundreds of TCR and CAR T-cell-based therapies are in late-stage preclinical development or clinical trials, such as the ones listed here that are sponsored by some of AllCells' HLA customers. The majority of HLA-restricted therapies are designed to accommodate HLA variants that are of high frequency. Some tumor antigen targets, such as the MAGE-3 antigen, are being tested in both CD4 and CD8 TCR therapies by utilizing high-frequency HLA class 1 and class 2 variants. You can see that two different MAGE-A3 therapies are targeted each to A0101 and DRB10401. The field of tumor immunology encompasses many applications that require the use of HLA-specific primary cells, from the identifying tumor antigens to generation and expansion of tumor antigen-specific T cells ex vivo to development of MHC tetramer for use in diagnosing cancer antigen treatments. Some of our most frequent HLA customers use large-scale HLA-typed blood collections to optimize manufacturing protocols for TCR therapies about to be tested in clinical trials. We will now move on to describe two more applications for which HLA is extended from the clinic to the bench in terms of understanding associations of specific HLA molecules with disease or response to therapy. Plummeting sequencing prices have facilitated enormous numbers of disease association studies dedicated to identifying genomic variations associated with disease. Both class 1 and class 2 HLA alleles have been associated with a variety of autoimmune and infectious diseases, and when used in this sense, constitute disease-related biomarkers that can be used to determine an individual's propensity for disease. HLA polymorphisms have been correlated with disease risk, immune response to infectious disease, as well as adverse response to therapies. Certain HLAs can facilitate disease or predict recurrence. Despite the large number of significant associations between HLA and disease, the most of any gene family in the genome, the functional basis for these associations are not well understood. Not surprisingly, researchers studying the mechanisms underlying such diseases, particularly autoimmune diseases, are interested in the HLA background of their primary cells. Some researchers simply want to control their results by limiting the HLA diversity in their primary cell experiment. Others want HLA-matched tissue as healthy controls for studies in diseased cells. Such strategies, by incorporating relevant genetic data into experimental design at all stages of research, can only improve our understanding of donor-to-donor and thus patient-to-patient variability. Multiple genetic and non-genetic factors can modify the action of a drug, resulting in a varied response to a particular drug across different individuals. Many adverse drug reactions are caused by an unexpected immune response, often referred to as drug hypersensitivity or immunogenicity. Genetic association studies have identified strong linkages between specific HLA class 1 and class 2 alleles and the immunogenicity of drugs, including FDA-approved therapies for diseases such as gout, epilepsy, and HIV. A notable example of the role of HLA in drug immunogenicity is abacavir, an antiretroviral drug approved in 1998 by the FDA that is frequently used in combination therapy to control HIV infection. Soon after the drug was released, it was discovered that approximately 8% of patients taking the drug exhibited symptoms of hypersensitivity, including rash, fever, and gastrointestinal distress. Genetic association studies have demonstrated a very strong linkage of immunogenicity in patients with the HLA-B5701 allele. The relationship is so strong that in 2008, the FDA relabeled abacavir to include a recommendation for testing for this allele prior to starting the drug. Many clinical studies now implement HLA screening to prevent or limit hypersensitivity reactions during drug trials. Due to drug toxicity incidents, not only with abacavir, but high-profile cases in which several monoclonal antibody therapies triggered cytokine release syndrome and sepsis, immunogenicity screening has become routine at every stage of therapeutic development. Preclinical in vitro studies are incorporating HLA genetic data in order to screen for candidates that do not interact with high-risk HLA alleles or trigger a hypersensitivity response by any other mechanism. In 2014, the FDA released this statement recommending the use of HLA mapping studies to help define subsets of the patient population at increased risk when investigating a therapeutic protein product. We have only begun to scratch the surface of applications of HLA data. Some other common uses are listed on this slide. As more associations are made between HLA and disease, this list will most certainly continue to grow. As you may now appreciate, the complexity of HLA's role in normal cell function, disease, and in cellular therapies is high. It is not surprising to talk with customers who are both immunologists and non-immunologists who struggle with interpreting HLA genotypes or understanding why a specific HLA type might affect their results or affect their data. We are here to aid you in that struggle. For those of you unfamiliar with AllCells, we are a provider of fresh and frozen primary hematopoietic cells and tissues, supplying the biomedical community for 18 years. A few years ago, we saw an increase in demand for HLA-typed cells and tissue, and we invested in a commitment to form comprehensive HLA typing, a large subset of our reliable donors. This investment has allowed us the ability to offer prospective collections, dramatically reducing lead times for HLA-specific fresh tissue. Our donor clinic currently manages over 200 HLA-type donors that are eligible for leukopak, whole blood, and or bone marrow donations. We also have in our frozen inventory over 100 lots of HLA-profiled MNC CD34 positive as well as T-cell and B-cell subsets. We can offer tissues and cells that are characterized for other immune genetic markers such as KIR or Fc gamma receptor. When you receive your HLA-type fresh and frozen cells from AllCells, you will receive high-resolution comprehensive data for 8 Class 1 and Class 2 HLA alleles, a sample of which you can see here. With the HLA primer that you have just received, you should be better prepared to digest and apply this information to your preclinical results and its interpretation. I hope that this webinar has clarified some of HLA's complexity and inspired you to explore how HLA might play a role in your research, whether as an experimental tool or as a way to control for and interpret donor-to-donor variability. Please don't hesitate to reach out to one of these email addresses after the webinar with follow-up questions. We are always very interested in hearing about your HLA applications. Also, if there are other genetic profiles you would like to see in your primary material, please let us know. It is only with your feedback that we can be prepared to meet your genetic and other customer requirements. Thank you all for your time. Afshi?
Thank you very much, Erin. Really enjoyed the informative presentation. Before we jump into the Q&A session, I wanted to point out a few things to the audience. Today's webinar will be posted on our website later on. In addition, you can find all of our previous webinars at the URL address listed on your screen currently, allcells.com/support/webinars. For any further follow-up, as Aaron mentioned, please reach us at the special email address we've set up, hla@allcells.com. We'd be happy to address your inquiry then. We will now begin answering any questions you may have. As a reminder, please enter your questions in the chat prompt to the right of your screen. Let's go to our first question now. Can AllCells provide HLA-matched and HLA-mismatched cells and tissues? Aaron?
So I think I implied at AllCells we use next-generation sequencing to type our donor material. With that level of data, we are able to provide matched and unmatched material. It's much easier for frozen than it is for fresh tissue, but we can do it for fresh. It probably would just require a lot of lead time. I would note, though, that matching can differ according to the person saying it. And before you request matching, I would suggest that you understand which alleles you would like to match, as it can range anywhere from 2 to maybe 8 of alleles matching, and that will help us get you the exact match setup for your needs.
Great. And kind of along those same lines, another question about the specific alleles. Is there a difference between A02 and A0201 alleles?
So as I mentioned briefly at the beginning of the talk, A02 is really only an allele group, and it encompasses A0201. So A0201 is a more specific allele than the classification of A02. A02 could also encompass A0203, that allele group. So it's just a level of resolution and specificity, and more we're seeing customers request the actual allele level data rather than the allele group. It allows for better control and tighter control over more specifically the tumor antigens that are being expressed for these TCR therapies.
Great, thank you, Aaron. Our next question is more about our donors and our donor pool. Is it possible to distinguish between HLA homozygous and heterozygous donors?
Yes, it is. Unlike with serology, sequencing techniques can reveal the multiple alleles at a given loci. So we are able to resolve whether or not a donor is heterozygous and homozygous. However, as you can imagine, with the level of resolution that you're seeing, it's very rare to find a homozygous donor, and so that's very hard to come by. So most researchers are happy with just having one allele, so a heterozygous positive for the HLA variant that they need for their research.
Great, thank you, Aaron. Looking to the audience for any further questions, and not seeing any. Okay, great. That concludes our webinar for today. On behalf of Aaron and the AllCells team, I'd like to thank you for your time and interest today. We look forward to hosting more educational presentations for you in the future. Thank you. Have a great day.